Today in this module, we will discuss an important topic which relates to the practical aspects of molecular biology, and that is making the antibodies. In this module, we will discuss the making of the polyclonal antibodies. Now, antibodies can be made in various ways, and the choice of which method is used is dependent on final assay format. Because antibodies are important components which are used in the assay of the antigen. Now, qualities of antibodies, that is to determine its usefulness in the method to be used. That whatever method we want to use, then its qualities are determined. And the one is that it can be defined specificity, that it should be very specific. And number second, its affinity and avidity. That is, its avidity is a property that how much active it is to determine the antigen. So these are the two which control the qualities of the antibodies. Now, polyclonal antibodies are raised in appropriate donor animals. And the donor animals, they are generally rabbits for smaller amounts. Then sheep or goats can be used for larger quantities of antibodies. Occasionally, rats or mice, smaller animals, they can be used for small research quantities of antibodies naturally. Now, adjuvants, these are the substances which increase the immunogenicity of the antigen. That the immunogenicity of the antigen, that is raised or that is increased by chemicals which are known as adjuvants, and adjuvants are used to reduce the amount of antigen required. That when we are using adjuvant, then the amount of antigen which is required is reduced. That is their advantage. Adjuvant may be the chemicals, that is the detergents and oils, and the complex proprietary products which contain the bacterial cell walls. So these are the adjuvant which they are made. Now, how we can make the antibodies? That the antibodies which are pre-immune blood samples, they are taken to provide the baseline Ig, that the immunoglobulin G level. Ig stands for immunoglobulin and G is the antibody. So what we do, that one is that we use the pre-immunization bleed, that is the before immunization, that is the bleed and that is tested. Then the animal, it is immunized first day. First immunization is on the 0 day. Then the second immunization is after 14 days, and so on. The third is after 44 days, and after 60 days, the test sample that is taken, and then the test quality of the serum by ELISA is made. If it is good, so immunoglobulin or the antibodies, they are screened. If not, again the process go back and to the 66 days of the animal immunization. Usually a range of doubling serum dilution, they are used. That definitely when we are using the antibodies, their concentration, that is determined, and the range of doubling serum dilution they are made, that is 1/100 times, that is made dilution 100 times, or it is further diluted to 1 over 800 times, 12,800 times, which is extremely diluted, and again tested against the antigen to which we are using it. Serum from a satisfactory course of immunization will detect antigen at about 1 over 6400 dilution time. So you can imagine that how small quantity of antigen can be detected. This indicates the high level of circulating antibody, that the antibodies level which are very high than 1 over 6400 dilution, up to that antigen can be checked. Once high level of circulating antibody is detected in test bleeds, then the donation can be taken. That is, what does it mean? That when it is 1 over 64,000 times diluted, the antigen can be detected, then the animal is bled and its blood is taken. Now, blood donation, they are allowed to clot, and the serum, this is collected. Then the serum can be stored at 4 degrees centigrade at low temperature or lower for longer period. If you want to keep it for longer period, then you have to store at 4 degrees centigrade. It is also possible to produce antibodies in chicken eggs. That is, it is not necessary that you use the bigger animal. You can use the chicken eggs, and immunoglobulin Y, that is IgV avian, that is the immunoglobulin is known as immunoglobulin Y, IgY, and the chicken, they secrete it into the eggs to provide the protection for the developing embryo. So by the eggs, and this can be utilized for effective polyclonal antibody production. That is, it can be used. Now, how? The chicken are immunized 3 or 4 times with the antigen, and then eggs are collected and can yield up to 50 milligram antibody per yolk. So we can use the chicken as well to prepare. The antibody has to be purified from the egg yolk. From the egg yolk, you have to purify the antigen which is produced, and the small quantities of very pure polyclonal antibodies can be produced in rats and mice as well in the ascitic fluid. Ascitic fluid is, as a matter of fact, it is a mammalian response to a tumor within the peritoneum. In the peritoneum cavity, that fluid is made, and animals are immunized with the antigen of interest. Once a high serum level is detected, then ascitic fluid production is induced in the animals. Now, non-secretory myeloma cells, that is another type, such as NSO, are introduced into the peritoneal cavity of the animal by injection and allowed to grow there. And it makes tumor cells. The presence of the tumor cells, it causes the animal to produce the ascitic fluid, which contains high levels of immunoglobulin to the original antigen, and that ascitic fluid is, as a matter of fact, drawn from peritoneal cavity, and then the further antigen or antibodies, they are purified from them. So you see that in this segment, we have discussed the various ways to produce the antibodies, how we can immunize, what type of animals we can use if we need small, and all that.